Dear students, I hope you all will be fine. I'm here, Muhammad Jamal Khan, lecturer in botany, University of Education, Lahore. Today, we will discuss a topic from one of the core subjects of botany, that is plant systematic anatomy and development. The course code of this subject is BOTN 1112.
So, in this lecture, we discuss the chemical composition of cell wall. So, here we begin. Actually, this lecture is the extension of previous lecture. As you already studied in the previous lecture that the cell wall consists of 3 layers. These 3 layers are primary wall, middle lamella and secondary wall. So these 3 layers are composed by different chemical constituents. That contains pectin, cellulose, hemicellulose, protein and other organic compounds. The arrangement of cellulose in cell wall is based on specific pattern. As you know, the cellulose is a polysaccharide, and the specific pattern of its arrangement by repeating molecules form different compounds. It may be a microfibril, it may be a bacillus, and it may be a macrofibril. Depends upon the arrangement of repeating units of polysaccharides. So the main theme to convey this is that we further extend the cell wall with reference to their chemical composition in this lecture. So here we have the chemical composition of cell wall. In this slide we mention different constituents related to the formation of cell wall. Basically, cell wall is composed of different chemical constituents and compounds. Here we have these chemical compounds that includes cellulose, hemicellulose, pectin substances, lignin. Then we have gums and mucilage, silica and calcium carbonates, cutin, suberin and waxes, and in the end we have other organic compounds. The most common compound in plant cell wall is the cellulose, that is basically the carbohydrate. So it's the repeating arrangement of carbohydrate molecule, that is polysaccharide. So basically cellulose is a polysaccharide. In woody plants it is impregnated with lignin, and cellulose is a relatively hydrophilic crystalline compound having the general formula, that is C6H10 and O5, and indicates the number of cellulose molecule. So it is a homopolysaccharide consisting of glucose molecules in chain. Cellulose itself is a polymer of round about 1000 to 10,000 glucose residues. This cellulose is the major component of the primary and secondary wall, and the cellulose polymers associate through hydrogen bonds as indicated in this slide. The association of hydrogen bonds results in the form of microfibrils and macrofibrils, which in turn interact with each other to form fibrils, that is also called as fibers, as indicated in this slide. So due to highly precise and orderly arrangement of such molecules, that is basically the architecture of cell wall, develops a crystalline properties of cellulose. As a result of this orderly arrangement, that results to develop crystalline nature in cellulose, cellulose chains form a structure that is called as micellus. One micelle is composed of 100 chains of cellulose, and micellus is considered to be the smallest structural unit of the cell wall. Hemicellulose are also the constituent of cell wall. This is a heteropolysaccharide or that is composed of variety of sugars that includes xylose, mannose, arabinose, glucane and galactans. This hemicellulose becomes highly hydrated and form gels. It is most abundant in the primary wall and is also found in the secondary wall. The yellow cylindrical structure in this slide indicates a hemicellulose, that is the component of cell wall. And these hemicellulose made of variety of sugars, that includes xylose, that is also called as xylanes or xyloglycan, mannose, that is also called as mannans or mannoglycans, xyloglucans, and mixed linkage of beta glucans. So here we have the next substance that play a very vital role in the formation of cell wall, that is pectic substance. So actually these are very close related to hemicellulose, because in pectic substances a group of polysaccharides that is made up of primarily of sugar acids, and the major compound that is present in these pectic substances is a polymer of galacturonic acid. So they are important constituents of plant cell walls and the middle lamella between adjacent cell wall. So normally they are present in insoluble form, but in ripening fruits and in tissues affected by certain diseases they change into a soluble form, which is evidenced by softening of the tissues. So the pectic substance present in three forms. One is pectin, the second one is pectic, and the last one is propectin. So one by one we discuss all these components. First of all, we have pectin. It is the major component of middle lamella, but also found in the primary walls. It is a polymer of around 200 galacturonic acid molecules. This pectin is less hydrated as compared to pectic acid. It is soluble in water only in one condition, when water is hot. The other one we have is pectic acid. It is also the major component of middle lamella and also found in the primary wall. And pectic acid is the polymer of about 100 galacturonic acid molecules. It is hydrophilic in nature, and it forms a salt bridge between calcium and magnesium ions. The next one is protopectin. Simply, we say that the protopectin basically a pectin, but it is a group of water insoluble pectic substances that occur in plants and yielding pectin or pectic acid on hydrolysis. So here we have the structure of galactonic acid, that is the main component of pectin or pectic substances. So basically this galactonic acid is a sugar acid, an oxidized form of D-galactose. Majorly it exists as a polymer, polygalactonic acid, in pectic substances. As far as concerned with its structure, in its open form it has an aldehyde group at C1 carbon and carboxylic acid group at carbon number 6. Now the next chemical content in the formation of cell wall is lignin. So it is the most important wall substance, and the chemistry of this subject is imperfectly known. So basically this lignin is a class of complex organic polymers that form key structural material in the support tissues of vascular plants and algae. In lignin, actually lignin is the polymer of higher carbon content that distinct from the carbohydrates. The major compound that is chemically present in lignin are the cross-linked phenolic polymers. This lignin actually is the end product of metabolism. So it is important in the formation of cell wall, especially in wood and bark. Due to this compound, it provides rigidity to the plant cell. Lignin is present in all three layers, as the middle lamella, primary wall and secondary wall. So due to this, it provides strength and rigidity to the cell wall. One term that is known as lignification, actually it means the impregnation or deposition of lignin in the wall is referred as lignification. So here we have the dramatical representation of lignin that indicates here in red color that is associated with cellulose and hemicellulose substances in the formation of cell wall. So the next chemical constituents are gums and mucilage. The gums or mucilage basically are complex carbohydrates or heteropolysaccharides of cell wall. They are related to the pectic compounds. Gum and mucilage have similar constitution and on hydrolysis yield a mixture of sugar and uronic acids. Mucilage are generally normal product of metabolism formed within the cell. The major function of mucilage is to store material, act as a water storage reservoir, and provide protection for germinating seed. Mucilage occur in some gelatinous or mucilaginous types of cell wall. As far as concerned with the gums consist of calcium, magnesium and potassium salts of polyuronides, and it can be hydrolyzed by prolonged boiling with dilute acid to yield a mixture of sugars and uronic acids. Gums are abnormal products formed by the injury of plant or during unfavorable condition like drought, or it can also be produced by the breakdown of cell wall. These gums and mucilage are carbohydrates in nature and are present in the outer wall of the cell layer of plant bodies of many plants. Gums are amorphous substances which are insoluble in alcohol and most organic solvent. It is only soluble in water. Due to this, it gives a viscous sticky solution. And some other the gums are swollen by absorbing water to form a jelly-like mass. While the mucilage is often found in epidermal leaf cells, in seed coats, roots and bark. So these are associated with certain types of cell like cells of algae and seed coat. Both the gums and mucilage substance swell in water. The next chemical constituent in the formation of cell wall is silica and calcium carbonate. Both these substances are the result of biomineralization, and biomineralization is the process by which living organisms produce minerals, often to harden or stiffen existing tissues, and such tissues are called mineralized tissues. So the silica and the calcium carbonate are majorly present in the cell wall of epidermis of grasses. In plant, the biomineralization is usually found at the level of individual cell as intracellular deposits. Most probably, the trichome tips of many plants are known to contain highly concentrated silica, while the calcium carbonates that are found as biominerals, concentrated deposits, or as composite minerals present in combination with organic cell wall material to provide stiffness to the cell wall. So these both compounds, silica and calcium carbonate, determine the mechanical properties of cell wall. So the next chemical substance that involved in the formation of cell wall are fatty substances. The most important fatty substances are cutin, suberin and waxes. In all of them, waxes melt easily, whereas cutane and suberin are not easy to dissolve. Cutane and suberin are closely related, highly polymerized compounds consisting of fatty acids. Cutane forms a continuous layer, that is the cuticle, on an epidermis, and the phenomena of impregnation of wall with cutane are called as cutanization, and the formation of cuticle is called cuticularization. Suberin along with cellulose occur in cork cells of periderm. The process of deposition of suberin is called as suberization. Waxes are deposited on the outer surface of many fruits, leaves, and stems. Some of the other substances like organic compounds have also a role in the formation of cell wall. So here we have first of all a tannin. Basically, a tannin compounds are widely distributed in many species of plant, and they have a role in protection from predation, and help in regulating plant growth. Tannins have different molecular weight range from 500 to over 3000 in case of gallic acid ester. So basically the tannin is responsible for the coloration or for the astringency and some of the flavor in plants. It normally occurs in root, wood, bark, leaf, and fruit of many plants, particularly in the bark of oak species. And it also occurs in galls as the pathological growth resulting from the insect attacks. The next we have the rhizins. These rhizins basically are viscous substances that convert into a rigid polymers through a curing process. Rhizin is typically the name to refer to a natural occurring organic compound that is sourced from plants. It usually consists of non-crystalline liquid substance that is fusible, making it an effective alternative in plants. Protection. So next we have volatile oil, that is also called as essential oil. So this volatile oil is a concentrated hydrophobic liquid containing volatile chemical compounds from plants. So these volatile or essential oil in the sense that it contains the essence of plants' fragrance. So the characteristic fragrance of the plant from which it is derived. Next we have pigments. Pigments are tightly bound to cell wall, and they have a long-term protective role. According to recent study, investigated that the pigments that are associated with the cell wall contributes to provide UV protection by plants, and from extreme habitat that enables the plant to survive. In the end, we have organic compound that exist in the form of protein. In cell wall, the protein occur in the form of hydroxyproline in the primary wall of various tissues. The amount present increases during growth. It is thought that the protein serve enzymatic as well as structural function in the cell wall. So this is all about for this lecture. Thank you so much.